These are the two parents. These are 3 children. Which one, if any, or more? So circle 1, none, or all, or more than 1. Which one of these could be the biological children of this couple? You do them in parts, right? Look at A first. Can you have an AB child? Clearly. Can you have an MN child? No problem. Because remember, these are independently assorted. Can you have a B child? A can be the father. This guy can be the father if the woman is heterozygous. And N easily, and N can come into play. This one is going to be a problem. Why? Because the B is a possibility, but the M is not. Because an MN, remember, is codominant with an N, so you cannot have an M child. So this one does not work. But really, this is all the major types of questions that will come from the first 2 chapters. Because remember, there's no epistasis, there's no sex-linked inheritance. So any questions from this? Now, 3 is mitosis and meiosis, and with 3, there will be some definitions, but it's largely going to be tracking the number of chromosomes and chromatids and non-disjunction and things of that nature. So what type of definitions would you be expected to remember? Right off the bat, you have to know the difference between, for example, the P and the QR. What is acrocentric, meta, submeta? What is a locus? What is a homologous chromosome? Right? Homologous, for example, true or false, and why? Homologous chromosomes are identical in every respect. Right? False, because they are identical in almost everything aspect, except not necessarily what? Alleles. You could have the same gene, you have to have the same gene, but it's not going to be the same allele necessarily. Big A, but it could easily be big A and little. As far as, you might get 1 or 2 questions, put these stages in order. PMAT, prophase, meta, ana, telo, right? Interphase is G1, GOS, G2. Don't worry about the details of prophase 1, leptonema, zygonema. Know the big milestone events, if you like. So for example, what happens in interphase? The chromatin condenses. S is replication. Prophase is what? Everything is condensing, the nucleus is falling apart. Don't worry about pro-metaphase. What happens in metaphase? They line up in the middle. Anaphase, they separate. Telophase, reverse of prophase. Cytokinesis, cleavage furrow, or cell plates. What happens in meiosis 1? It's more elaborate. The hallmarks of meiosis 1. What happens in meiosis 1 that does not happen in meiosis 2? 2 big events. One is crossing over. The other one is exactly tetrad alignment randomly. And that is what genetic diversity is all about. It's crossing over and independent assortment, which is related to independent alignment, really. So let's use this key and try to get mitosis and meiosis out. 2n gives you 2n. 1n gives you 1n. 2n gives you 1n. 1n gives you 2. Which one of these best describes mitosis in humans? Which one of these best describes mitosis in humans? Right, clearly A. Diploid gives you diploid, there's no change. Which one describes meiosis 2 in humans? Meiosis 2. Meiosis 2, you should remember, is which division? The equational division. Which means what? That you do not reduction division. Okay. What is meiosis 1? Meiosis 1 is where you go from diploid to haploid. C. Meiosis 2 is B. You're keeping the haploid haploid. Right? You can circle more than one or none. Well, actually, we've already circled one. So, but if the question was, which one can describe mitosis in general, what would the answer be? Not in humans. Mitosis in general. A and what? A and not D. A and B, right? because mitosis can happen in yeast cells. In yeast cells, they start out as haploid and they continue as haploid. Remember, you don't have to know spermatogenesis. You do have to know bar bodies. Okay? And with bar bodies, what is a bar body? How many bar bodies are there in a particular instance? Remember, it's the number of x's minus 1. And you have to be able to distinguish how does DNA dosage compensation occur between the difference between, for example, Drosophila and Sinorhabditis elegans versus humans, right? In humans, one X is inactivated. In Drosophila, the X in the male is hyperactivated. And in Sinorhabditis elegans, the two X's in the hermaphrodite are half activated.
Can you say that one more time? Sorry.
So, it's in the notes at the end, but the difference is that in Drosophila, the X is hyper. In humans, one X is hypo, if you like, is inactivated. And in Sinorhabditis, both X's in the hermaphrodite are hypo, are less. And you'll be able to work out simple problems with calico kittens, right? For example, a calico, true or false, all calico cats are female. False, right? For you to get a calico male, it'll have to be what? It'll have to be a Klinefelter cat. It has to be 2 X's and 1 Y, and they are found, right? 2 X's and 1 Y automatically means they could be heterozygous, and that's the whole idea, right? So if you have, let's say, a calico cat times an orange cat, what's the probability of getting a calico kitten? If you have calico times orange, remember, calico is orange and black. That's calico. Times orange and what? Why? Because males, no breaking news here, not males. Cats are mammals. XY system. XO, XOY, XBO, XPY. There's only one calico. 1 out of 4. And remember the terminology. If it's asking you what percentage of their kittens is going to be calico, it's 25%. What percentage of their female kittens will be calico, and that's 50%. So, any